Howdy, my name is Mikaela Sebastyn, and I am a second-year veterinary student at the Texas A&M College of Veterinary Medicine and Biomedical Sciences, and today, on behalf of the PEER program, we are going to talk about genetics. The study of genetics began with observations made by Gregor Mendel, who lived as a monk and had a strong interest in science. After noticing that the flowers of his pea plants were either violet or white, Mendel began to study the segregation of heritable traits. Between 1856 and 1863, he cultivated and tested at least 28,000 pea plants. Remember that Mendel worked almost 150 years ago when nobody knew about genes or even the structures that carry genes. Traits that Mendel observed in his pea plant experiment are depicted in the image below. He studied seed shape and color, flower color, pod shape and color, flower position, and stem length. Let's consider a single gene. A gene carries information that determines your traits. Traits are characteristics you inherit from your parents. Genes are located in chromosomes, which come in pairs and carry thousands of genes, as depicted in the picture on the right. More specifically, chromosomes are structures in the nucleus of the cells that consist of DNA. But did you know that there are two types of chromosomes that determine an organism's gender? There is an X chromosome and a Y chromosome. What do you notice about them? If you guess size difference, you're not wrong on that. Let's look into these chromosomes a little bit more. As mentioned before, X and Y chromosomes are important for determining gender. You have to have a combination of two, either an X and a Y for a male organism or two X's for a female organism. Like all genes, you get these from both your parents. Because females are XX, you would only have one X from your mom. And because males are XY, you would either have an X or a Y from your dad. In humans, a cell's nucleus contains 46 chromosomes, or 23 pairs of chromosomes. Half of the chromosomes come from one parent and half from the other parent. The image on the right is an example of a normal human karyotype from a male. A karyotype is a visual layout of all the chromosomes in the cell of an organism. Notice that there are two of each type of chromosome, as well as the X and Y chromosome. The role of a gene is to control a trait. A trait can be anything we visibly see on someone else, such as hair color, eye color, and sometimes fingerprints. Ever heard of how each person has a unique set of fingerprints? Well, guess what? Even twins have different fingerprints. If you lose the skin on your fingertip, in the case of a cut or a burn, it will grow back in the exact same print. Do this right now if you can. Rub a pencil over your fingertip and touch it to a piece of paper. This image depicts the different patterns of human fingerprints. Take a moment to pause the video and compare your fingerprint pattern to one of the patterns shown. If you've ever felt lost or confused in biology, now's the time to relearn. If biology is a breeze for you, then you're in for a treat. Heredity is simply known as the passing of traits to offspring, as is the case with you and your parents. This process often involves one becoming predisposed to characteristics of its parent. For example, if you have a family history of heart disease or diabetes, there is a chance you may develop such problems in the future. An allele is a discrete version of the same gene. That is, if your mom has brown hair and your dad has blonde hair, you inherit the alleles for both, but you only express one hair color or the other. Genotypes are the genes of an organism for one specific trait, while phenotype is the physical appearance of a trait in an organism. Dominant trait refers to a genetic feature that hides the recessive trait in the phenotype of an individual, whereas recessive describes a trait that is covered over by another form of the trait and seems to disappear. Going back to the hair color example, if you have brown hair, your mom has brown hair, and your dad has blonde hair, that means the allele for blonde hair you received from your dad is most likely recessive, and the allele for brown hair from your mom is dominant. Now that we've discussed alleles, we can now use the following terms to describe an organism. Homozygous means it has two of the same alleles for a trait, otherwise known as pure, and heterozygous means it has two different alleles for a trait, or also known as hybrid. Your mind is probably spinning at this point with so many definitions to keep in mind, so let's break things down a little bit more. Remember that when we study the potential outcome of various offspring, a capital letter represents the dominant form of the gene, and a lowercase letter represents the recessive form of the gene. As mentioned before, a genotype is a set of genes for a trait, so two alleles are needed for every trait. For example, a genotype is always written as big A, big A, big A, or little a, little a. The letter represents the trait, such as eye color or height. A capital letter means the trait is dominant and can be expressed as either big A, or big A, little a. Recessive can only be written as little a, little a. Remember the difference between homozygous and heterozygous? Remember, homozygous has two of the same alleles for a trait, while heterozygous has two different alleles. In this example, we can combine terms and say that big A is homozygous dominant, big A is heterozygous, and little a is homozygous recessive. A pedigree chart is the chart that shows all of the known phenotypes for an organism and its ancestors. For those of you who own dogs, sorry, it's not related to your pet's food in any way, but it does show us how traits are passed down and why we look the way we do. Let's look at an example involving a dominant disease. Let's say the dominant allele, or the big R for the disease, causes a dog to have an extra tail. In this case, the unaffected father has a homozygous recessive genotype, or two little r's, and the affected mother has a heterozygous genotype, with one big R and one little r. 4 puppies are born to this set of parents, which results in 2 of the puppies having the same dominant disease with extra tails, and the other 2 of the puppies are unaffected by the trait. Note the genotype of the affected puppies compared to the unaffected puppies, and think about each of their phenotypes. Now let's look at another example, but this time a recessive disease. Let's say the father is a carrier of an allele for a recessive disease, or little r, and the mother is a non-carrier, meaning she has 2 big R's. To be clear, a carrier means an organism has one recessive allele for a disease, but it does not express it because it is covered by the normal dominant allele. If 4 puppies are born to this set of parents, there is a great possibility that 2 puppies will inherit the abnormal recessive gene, and the other 2 puppies won't. Therefore, the puppies who do not inherit the abnormal gene will not be carriers of the abnormal gene in the future. None of the puppies are affected by the disease because they do not have the homozygous recessive genotype, or 2 little r's, where the disease would be in full effect. Again, notice the puppies' genotypes and think about each of their resulting phenotypes. Another way we can study genetics is by using Punnett squares. This is the standard method of predicting what the possible offspring of two parents will be. It is a helpful tool to show allelic combinations and predict offspring ratios. Before we go on further, let's review how to set up a Punnett square. You can try this at home right now. Begin by constructing a grid of two perpendicular lines. Next, put the genotype of one parent across the top and the other along the left side. For this example, let's consider a genotype of big B crossed with little b. Notice how only one letter goes above each box, but it does not matter which parent's genotype goes on either side. Next, fill in the boxes by copying the column and row head letters down and across the empty spaces. If we look at this Punnett square, you'll see how 4 boxes resulted in heterozygous offspring when we cross a homozygous dominant parent with homozygous recessive parent. Now that you've learned the basics of genetics and the Punnett squares, let's put your knowledge to the test. Let's say in this example that little w is recessive and results in a violet flower phenotype. Pause the video and make your own Punnett square to determine the percentage of offspring that will have violet flowers. The parents in this cross are heterozygous. Now let's look at the results. Your Punnett square should look something like this. Were you able to figure out what percentage of offspring have violet flowers? If you guessed 25%, you are correct. Let's look at another example. Red hair, big R, is dominant over blonde hair, or little r. Pause the video and make a Punnett square between heterozygous redhead and a blonde. Your Punnett square should look something like this. Can you tell by looking at it what percentage of offspring have red hair? The correct answer is 50%. Let's try one more example. Black eyes, or big R, are dominant over red eyes, or little r, in rats. Pause the video and make a cross between a homozygous rat with black eyes and a rat with red eyes using a Punnett square. Your Punnett square should look like this. What is the possibility of red-eyed offspring? If you guessed 0%, you are spot on. This concludes our presentation on genetics. Thanks for watching.